Hello everyone, my name is Nurin Akila binti Nosham. My metric number is E2A0175. Today I want to talk about introduction, genome size, accession number and supplementary table 1 of laboratory. Firstly, about introduction, Hevia brasiliensis is a organism of laboratory, is a perennial crop of Euphorbiaceae and indigenous to the Amazon basin in South America. The domestication of rabbitry has contributed greatly to the human lifestyles in the past 150 years. The rubber tree provides more than 98% of the total natural rubber production worldwide, although there are 2,000 rubber-producing plant species including Parthenium argentatum, Tarazacum coxsackie, and Euphorbium ulmoides. Secondly, about genome size. Genome size was estimated to be 1.50 until 1.62 GB based on the 17-mer to 27-mer distribution analysis. This can refer to supplementary table 1. Third, about accession number. Published transcriptome sequencing data by Gene Expression Omnibus (GEO), which is GSE 80596. The table shows about supplementary data 1, which is global statistical comparison between CATAS879 assembly and available laboratory genomes. There are 8 differences in the table, which is assembled genome size, number of contigs, N50 contig length, number of scaffold, longest scaffold, gap number, LTR/copia, LTR/Gipsy. There are one URL in this study, which is NCB.
List of organisms closely related to the rubber tree. The rubber tree is closely related to several species within the Euphorbiaceae family. Among the list of organisms provided, only Manihot, Esculenta, Crus, and Ricinus communis L belong to the Euphorbiaceae family. The rest of the organisms belong to different plant families. Here is a phylogenetic tree illustrating this relationship. I used protein blasts, then this tree was produced using blast pairwise alignments.
The original research paper that published this genome, Rahman et al., draft genome sequence of the rubber tree Havia brasiliensis. Phenotype
Next is the bioinformatics tool used to analyze the genome. There are a total of 15 analytic methodology used in this study, which is the first one is the genome library construction and sequencing. The second one is the high-C library construction and sequencing. The third one is the bio-nano genome map construction. These are all the three methodology flow mentioned earlier. The first one on the left is the genome library construction and sequencing. In the middle is the high-C library construction and sequencing. And the third one on the right is the bio-nano genome map construction. Next, the fourth one is the de novo genome assembly and validation of Catas-X79. And the fifth one is the RNA extraction, transcriptome sequencing, and isosequence. The sixth one is the genome annulation, and the seventh is the gene family and synteny analysis. Eight is the mapping of genome reads and variation calling, and then ninth is the population genetic analysis.
Phenotyping and Genome-Wide Association Study (GWAS). The study was conducted on 208 10-years-old Havia accession, including 93 cultivated and 115 wild germplasms. The bulk of the tree was baked in ethanol treated with iodine and bromine and abandoned in paraffin. The NLR was counted under ELISA DMLB microscope. Genome-Wide Association Analysis (GWAS) was conducted using the linear mixed model algorithm in the Emacs program. A kinship matrix was generated to correct population stratification. RNA isolation, cDNA synthesis, semi-quantitative, and QRT-PCR analysis. Total RNA were extracted using the RNA prep per plus kit, and its integrity and quality were evaluated. Approximately 1 UG of total RNA was synthesized using a Revert 8 first-strand cDNA synthesis kit. Semi-quantitative PCR was performed, and QRT-PCR was performed using the CFX3X4 real-time PCR system. Relative gene expression level were normalized using 18S RNA, HBUB, C2b, HBUB, C3, and TKGA PDH. Three biological replicates were conducted. Yeast 1 hybrid assay. The yeast 1 hybrid assay involved cloning HBPSK5 from a region into the PABI vector and 29 HBMYC CDS into the PGADT7 vector. This vector were co-transformed into Y1H gold strain with PGADT7, REC P53 and P53 ABI as positive control and PGDAT7 and PHBPSK ABI as negative control. Transient dual luciferase assay. The study involved cloning CDS of 5 HBMYCS into the PCAMBIA 2301 effector vector and a reporter construct of HBPSK5. The vector were cultured in A. tumefaciens and infected onto healthy leaf of Nicotiana bintamiana. The LUC and REN luciferase activity were detected using a dual luciferase reporter assay system with 80 independent biological replicates performed for each combination. Electrophoretic mobility shift assay EMSA. The study used the electrophoretic mobility shift assay kit to analyze the binding relation between HBMY C18 and HBPSK5. The CDS of HBMY C18 was cloned into PET28A and transformed into BL21. The recombinant HBMY C18, his protein was expressed and purified. The DNA protein complex was separated on polyacrylamide gels and captured using an Azure Biosystem C300 imaging system. Functional identification indirect circumcoxeglycerin. The study cloned plasmid 35S HBMYC26 and 35S HBPSK5 into the PFGC5941 binary vector and introduced them into Agrobacterium tumefaciens strain AGL1. The phenotypes of TO transgenic line were examined after tissue culture. Root examples from 3 months old plant were fixed in ethanol for 24 hours, sliced via a victrum, stained with oil red O, and mounted in glycerol. The lenticular cells index was used to estimate the abundance of lenticular cells in tissues.
The tool and methodologies mentioned their advantage and disadvantage. First one is allow for understanding the genetic diversity and structure of rubber tree population. Analyzing the genetic makeup rubber tree population help conserve resource, manage breeding, and ensure sustainability. Determine the genetic connection and origin between white population and domesticated groups. Reveal relationship and origin aiding interest in domestication and introducing valuable traits from white population. Provide a measure of genetic variation within population. Measuring nucleotide diversity indicates genetic variation important for disease resilience and environmental adaptability. Useful for assessing the impact of domestication on genetic diversity. Allows for understanding the molecular mechanism underlying important traits like lattice production. Advanced genetic and genomic tools provide deep insight into the domestication and improvement of rubber tree. Tools like genetic and LD analysis reveal region under selection and guide breeding improvement. Hormonal signaling studies open up avenue for genetic and biochemical intervention to enhance rubber production. The first one of these advantage is require large dataset to detect significant linkage disequilibrium pattern. Interpretation can be challenged if LD extend over large genomic region. Hormonal signaling pathways are often complex and can be involve numerous interacting components. Dissecting this pathway requires sophisticated experimental technique and can be technically challenging. Functional studies can be
time-consuming and require precise experimental conditions. Gene function in model organisms may not always translate directly to fight conditions.